This is preparation for lecture 7. Um, the fly work is starting to get more complicated, so I'm going to try to really point out the key things that you need to remember. I hope this is not showing up because it won't go away. I keep trying to get rid of it. There. And so we introduced the idea of maternal effect genes and zygotic genes and just some basics of fly development. And I finished off talking a little bit about bicoid. And we will finish just the maternal part of the anterior-posterior patterning in flies. And I'm just going to have one quick description of the terminal patterning, because the very tips of the embryo, the anterior-most and posterior-most, have their own special little system for determining and specifying those structures, and then dorsal ventral patterning. And here we'll look at both the maternal part and the zygotic part, so some of the zygotic genes that are expressed in response. So, and it is starting to get a little more complicated, so this is a little bit on the long side. Okay. Okay, that's not what I want. There we go. I was trying to get that menu to go away. So we'll talk about bicoid, and it depends on how busy the class was last time, where we got to, but I'm recording this a little in advance. So this may be review or it may be new. Bicoid is one of the first maternal effect genes that was identified. Bicoid means two tails, and the mutation was it's a maternal gene, so the mutant embryos come from mothers who have the mutant phenotype, so they are bicoid mutant mothers. And this is sloppy and it can be confusing, but it's not the genotype of the embryo but of the mother, and that's something to keep in mind. But so the embryos, this is a wild type fate map. The telson and the acron are the most anterior and posterior terminal structures. So this is the telson here, this little bit, plus the respiratory appendages at the back. The acron is just before the head parts of the embryo, and there's this little micropyle here as one of the structures. And when there are eggs laid by mutant mutant mothers, even if the embryo's genotype itself is normal, the mother has failed to provide the bicoid, and because of that, the embryo is not able to develop normally. It has two telsons, one that shouldn't be there and one that is in the normal position. And you can't really tell from this picture, but the actual pigmentation on some of these segments, these are the little segments of the embryo, are also somewhat abnormal. So we're missing head structures, and we have a duplication of the tail structures, the posterior structures. Now, if you look at the gene, if you look at a freshly laid egg, let's say, that's has been fertilized very recently, you will see the RNA for bicoid up near the anterior end. And it's actually fixed there. There's proteins lining the inner part of the membrane of that egg, and the RNA is glued basically to that. So the RNA can't, float around. However, after fertilization, the RNAs get translated and proteins are starting to be produced. And so you can see, I think pretty clearly here, that there's a lot of protein here, and less here, and even less here, so that when you get towards the posterior, there's none at all. So we're seeing a graded expression of that protein. And just as a reminder from, I think it was lecture 4, the features of a morphin are graded expression, so some sort of gradient, and then the ability to affect the gene expression and cell fate, so that in places where you have lots of bicoid expression, you have anterior structures, and where you have lower levels, you have maybe thoracic structures. That's where the legs and the wings are attached, and then when there's less, abdomen, and so on. So the idea being, if this is the morphogen, and we will explore that in the lecture, then you expect this graded expression to correspond with, think about the whole idea of the thresholds and stuff that you worked with after lecture 4 in that worksheet, that you have these different outcomes depending on how much of the expression is received by those cells. These are the kinds of evidence that we're going to look at. So the protein is found in the nucleus. It's stronger in the anterior. So it's kind of, it's in the nucleus at about the time we're expecting some specification of anterior versus posterior. It forms a gradient. When you make a mutation in it, it's lost the anterior part of the embryo. There are a series of injection experiments that you can use to rescue mutant phenotypes. This is one of the ways we find out what the important genes are in a process. If you and I'll talk more about that in the class, so I won't necessarily talk about it right here, but you'll hear more about rescue. And then if you overexpress bicoid, you get an expanded head region. And once again, go back to that gradient exercise in lecture 4 and think about how when you change the amount of the signal in that sort of theoretical model, you can change the relative proportions of the outcome of the different structures. And then if you put bicoid messenger RNA into embryos in the wrong place, like not up at the head, we should say these are embryos from mutant mothers. So they don't have any of their own bicoid. So if they're left on their own, they won't develop properly. But if you go ahead and inject some normal bicoid RNA, wherever you inject it, you're going to get head structures. And so those are the whole gamut of experimental evidence we'll go through in a little bit more detail in the class. And just think about it. Which of these pieces of evidence would be the most convincing that that bicoid is actually the morphogen, and what would you consider more circumstantial? So it could just be coincidence, right? Okay, so how that gradient forms, I've already mentioned. The RNA is affixed to the anterior inside of the egg, which at this point, remember, is just one big cell. And right after fertilization, translation begins, but it doesn't all get translated at the same time. It takes time. And so as the protein is being produced, some of it can start diffusing away from, it's now free, it's not stuck anymore. So it can go wherever. And again, we still have a bag of nuclei, but you see that there's lots of protein at the anterior end and less as you move towards the posterior. And so this is the representation of that. And at some point, cellularization is going to occur, and you're going to see then that graded image. And the other molecule we're going to talk about a fair bit is nanos. So nanos is the equivalent in the posterior. So it works together with bicoid. Bicoid graded from anterior to posterior, nanos from posterior to anterior. And if you look along the intersection of these two gradients, you see slightly different shades all the way along, which is meant to indicate different levels of relative amounts of bicoid versus nanos that all along the embryo from the anterior to the posterior basically provide information to the cells that end up there about what exact position they hold in the embryo, and therefore what structures and what kinds of tissues are going to be developed, and therefore what genes are going to be expressed. Let me just erase that dot. And so we're not going to go into the details about nanos experiments, but imagine if nanos works the same way. It doesn't quite work exactly the same way as bicoid, so you need to know that. But supposing that it was a morphogen in exactly the same way that bicoid is the morphogen, I would like you to be able to describe experiments you could do to demonstrate that, and what the result would look like. So how would you do the experiment, and what would it tell you? Okay. And this just shows that the process of making the gradients is the same. So you've got this big fertilized egg, and the nanos RNA is kind of glued to the posterior end, and then upon fertilization, the translation of that RNA begins, and we get this, the protein gradient from posterior to anterior, and then this is bicoid. I think the same picture or similar to the one before. Okay. And so what does bicoid actually do? It turns out that bicoid does two different things, and they're both really important. One is that it will activate the transcription of an anterior specific gene, hunchback. So hunchback is one of the earliest zygotic genes to be expressed. Zygotic means it is expressed from the DNA of the embryo itself, whereas bicoid came from the mother. It was put into the egg by the mother. But it does something else. You may remember lecture 3, I think, when I talked about the different levels of control of gene expression and how transcriptional repression is the best way of preventing gene expression because it's not wasteful, right? But this is an exception to that. Bicoid will bind to the mRNA of a posterior specific gene called caudal, and it will prevent it from being translated. So the mRNA is there, but if bicoid is bound to it, it can't be turned into protein. So you're preventing caudal expression in the anterior. And this turns out to be very important. So, and what's really interesting, excuse me, is that the hunchback gene is the first gene to be transcribed in the embryo, but mom puts a little bit of it into the egg. So if you look in an embryo, you're not going to see just, hunchback just in one little region. It's found dilutely in low amounts across the whole embryo. And then what nanos actually does, it's a translational repressor. So the hunchback messenger RNA is there. When the nanos protein is made, the protein kind of binds to the messenger RNA so that it can't be translated. So hunchback messenger RNA is found throughout the embryo in just a small amount. But wherever you've got lots of nanos, that protein is never produced. And so that makes only the front end of the embryo contains hunchback protein. And this is quite an interesting thing because the mother, instead of just putting hunchback only in the anterior, the mother puts hunchback all the way across, and then nanos has to prevent it from being translated in the posterior. So you have this extra little complicated way of regulating that expression. Okay, and so here's the sort of summary for both of them. If we look, how do we get these sort of protein gradients? You have, interestingly, hunchback and a gene called caudal. Mother loads into the egg messenger RNA for both of these, and it's evenly distributed. So you've got these across the whole embryo, you've got these two RNAs. And then there's this sort of complicated regulation of that because you've now got these localized maternal RNAs. You've got bicoid glued to one end, the anterior, nanos glued to the other end, the posterior. And then when those get translated, bicoid protein is made. Bicoid prevents, that's what this symbol means, prevents the translation of caudal. So caudal is, there's no caudal protein up in the anterior end, but it does activate transcription of hunchback. So you have the maternal hunchback plus a little more that's being promoted by bicoid now. So that means hunchback is a gene that is provided by mom, so it's maternal, and it's also transcribed in the actual embryo. So it's a zygotic gene, and it's one of the earliest zygotic genes to be expressed. And then nanos here is preventing hunchback from being translated. It doesn't have any effect on caudal. So caudal is kind of like, it's almost like a default in a way. Wherever there's no bicoid, then you have the translation of that protein, and you've got lots of it in the posterior and less of it as you move anterior. So in the proteins, all the proteins form these posterior to anterior or anterior to posterior gradients. But that is due to the localized expression of just 2 of those 4. Okay, so that's something you may need to go over a few times to get your head around it, because it's probably not something that's very familiar. And we will, of course, I'm going to go over it in the class as well. So you'll get another chance there, and I'll be asking you some clicker questions and that sort of thing to help you think about it. So the terminal system is right at the very tips of the embryo. And this is a case where you have a receptor, and the receptor is called torso. And if you were to look, it's probably useful to just have this. The egg membrane is the membrane that's inside. And then there's the vitelline envelope, or the vitelline membrane sometimes, that's on the outside. So you've got these 2 different membranes, and there's a lot of stuff, a lot of signaling and so on that happens between the vitelline envelope and the egg membrane. So there's this little place called the perivitelline space. And you'll see that in the text, and we'll talk about it in the class as well. And so what's interesting is you've got this receptor, and it's called torso, and it's on the egg membrane everywhere. And we just tend to, have these kinds of schematics. It's not really what the thing looks like. It's just a schematic. And so it's all around. But the mom has loaded the ligand for that just at the very termini, the ends, posterior and anterior, of the embryo, and those are being released and just there. And so although the receptors are all around, it's only in the very posterior or the very anterior regions that they get activated. And then there's a series of signals that then lead to the development of specific terminal structures. And so at this end, torso activation plus bicoid, which is anterior, is going to give you your anterior structures, and then the torso activation plus caudal. Caudal does most of the work of posterior specification, but nanos is needed to make sure that the anterior posterior is maintained. Okay, and really, I mean, there's quite a lot. It's quite interesting, and we'll talk when we talk about how the mother patterns the egg, probably, I think, next time, if I remember correctly. I haven't looked at those notes yet. But there, we can see how during the development of the egg, how those terminal structures are specified, how the ligands are laid down in particular regions. Because as the egg is developing, information has to be given to where the anterior, posterior, dorsal, ventral, and so on are going to be. Okay, so that's all you need to know for that part. And then we're going to finish off, this is going to be on the long side, but I want to talk about the dorsal ventral system. This is the first time we're really going to talk a lot about or at least more, about the actual zygotic genes too. But again, we're starting with the maternal system. And again, this close-up shows you the egg membrane, and this is the vitelline envelope. And just like in the terminal system, you have signaling across this space, and it's really, really tiny. The people who studied and first started isolating the proteins, the ligands and so on for these, one of them won an award for the work because it was so grueling. And he literally had giant pails of embryos, because each embryo, this is just microns across, this really tiny amount of actual fluid and space here. And so you needed to use massive numbers of embryos in order to be able to isolate this little bit of fluid and try to get the proteins, and then analyze the proteins and figure out what they all were. Huge amounts of work. Okay, so if you were to look along the whole embryo, the toll receptor is found throughout. So the toll receptor is everywhere around here, but only on the ventral surface do you have part of a ligand. There's a protein called Spätzle, which I'll talk about in a second, and it gets cleaved to form an active ligand. And that ligand then can diffuse across the little perivitelin space, binds to the toll receptor, and then that causes activation. And the activation leads to a protein called dorsal moving into the nucleus. And it moves into the nucleus only where there's, reasonable amounts of toll activation. So there's lots of dorsal protein in the nucleus at the very ventralmost surface. And then as you move up and around, the amount of activation, think the ligand is right here, so the chance of it diffusing way up here before it activates the receptor is less, and then the chance of it getting up here is even less until you get sort of halfway up, and there's pretty much no chance at all that ligand is going to make it around to that distance. And so what you have now, the gradient we're talking about here, is a gradient of dorsal protein in the nucleus. And dorsal protein is a transcriptional activator. It turns on a bunch of genes. And the genes it turns on are needed to form ventral structures, and then what we call ventrolateral. So ventral, but on the side, lateral. And then dorsal fates and dorsolateral are a different gradient. And that gradient is actually based on the absence of dorsal. So if you don't have any dorsal in the nucleus turning on certain genes, then your default is a gene called DPP. And this produces a secreted and diffusible signal. Okay, so Spätzle ligand down in the ventral side, it signals the toll receptor. A series of steps that we're going to talk about in a moment lead to dorsal going into the nucleus and turning on your ventral specific genes. But because you've got a limited amount of ligand here, the chances of it diffusing further up and around is lower and lower and lower, so we get this graded, where there's lots of signal, there's lots of dorsal in the nucleus, but then as we move up, when there's less signal, there's less. Some of the dorsal is still remaining in the cytoplasm. Okay, so that's the idea. And I'm showing you this picture because these are Spätzle. They are a type of German noodle. And the people doing the research called the embryos that were mutant for Spätzle. So the signaling didn't happen, and the embryo is dorsal all the way around, reminded them of this noodle. Okay. And if you have German background or any familiarity with this kind of little noodle, then it might help you remember. So here's a schematic. This is from the textbook, and then I'm going to show you another one and talk it through a second time just because I think it's helpful, and I'm going to point out a very interesting feature. So the toll receptor is a transmembrane receptor. It functions in dimers like this. Spätzle is a molecule that is cleaved by a protein called ester, and there's something else that then activates ester and so on. There's a series of steps here. I want us to try to keep it limited to just part of it, because all of this stuff is really complicated and you could easily get lost and confused. So I would like us to start here. A protein called ester cleaves Spätzle, and then when it's in the cleaved form, it's able to diffuse across and activate the receptor for toll. Toll is transmembrane, so the inside part of the receptor can bind to a gene called tube. There's another protein here that's kind of like an adapter. It makes a connection between tube and pele, and it's just called DMYD, DMID88. And so these three are in a kind of a complex. When toll gets activated, then pele is a kinase, but it phosphorylates itself. So it adds a phosphate to itself, which then indirectly leads to the activation of a kinase that phosphorylates a protein called cactus. These all have funny names because they're mostly named after what the embryos look like when the gene is missing or mutated. Okay. Now, cactus's job is to bind to this protein called dorsal and to keep it in the cytoplasm. It's a transcription factor, but if you keep the transcription factor in the cytoplasm, it can't transcribe anything, right? So then if you actually bind to, phosphorylate, and degrade cactus gets cut to pieces. Now dorsal isn't connected to anything, so it's able to go into the nucleus and it turns on a bunch of genes. Okay, so that's the procedure, and those are the steps we want you to know. This looks like a face. Okay, now I'm showing you this picture again. It comes from an article which I've cited down here. PubMed has lots of really great articles. And this is the fly version. So here's the spatula ligand, and it's binding to the toll receptor. And this is the internal part of the domains. They're just called TIR, T-I-R. And then this is the binding to the mid. Notice that this is on the other side in the other picture. There are some details that we're not 100% sure about. So I have found in different publications slightly different versions. So the key thing is to just remember that these three are all bound to each other. Okay, and then Pele is able to phosphorylate itself when the receptor is activated, and all the things are bound to each other properly. And then there is a separate molecule that then goes and phosphorylates the cactus. And this here is probably better than the circle that we were showing before, a more accurate representation of how cactus binds to dorsal. When it's phosphorylated, though, it gets broken to pieces, and then dorsal goes in to the nucleus and starts transcribing downstream genes. Now, what is interesting about this kind of signaling is not only does it create the dorsal ventral specification in the early embryo, but with just a couple of small differences, it also has immune functions. So flies have what we call innate immunity. They have built-in immune responses to certain kinds of pathogens, such as certain kinds of bacteria and so on. That's just already pre-existing, and they can respond to those and protect themselves using a mechanism that looks very much like this, except that the protein that goes into the nucleus is different. It's called diff, and when it goes in, the things it transcribes are going to be peptides and so on that protect the body against these bacteria, right? It turns out that humans have the same system, and the protein that they're recognizing is flagellin, which comes from bacteria, and they then go through, and you can see like the names of the things are different, but the way it functions is practically the same. In the case of human immunity, we know more of the between this part and this part. In flies, you know, we only have this. And in the human immune function, of course, it's been brilliantly well studied. So this shows you how this all works. But it's essentially the same pathway, and it works in you to protect you from certain kinds of bacterial pathogens and so on. So I throw that in there because it's nice sometimes to see flies and humans are so terribly different, and yet we have all these amazing similarities. And some of you may be super interested in human health, and especially if you've taken immunology or are going to, this kind of thing will be of interest to you. Okay, so basically where we've left that off with was just we've got this graded expression. We have lots of dorsal in the most ventral places, and then if you go up to the ventrolateral, we have less, and then we have little or none on the dorsolateral, and then the most dorsal is also different. And so this, as I said, the dorsal ectoderm DPP and tolloid are the two proteins that are being expressed there. The amnioserosa is this part up here. This is not actually going to be part of the embryo, but it's a source of some signals. So there's a gene called ZEN that gets expressed up here. You only need to remember the abbreviations. Tolloid, I'm pretty sure is TLD. Rhomboid is Rho. SOG, shortened gastrulation, SOG. And then twist and snail. These are mesoderm specific. And I think that's all you'll need to know is that they're mesoderm specific. But how do these things happen? How do we get, you started with this gradient, but you've got these nice boundaries, and part of that's going to be based on thresholds and so on. And so once those thresholds are set, how do we maintain these very specific domains? As I mentioned, I think I did, that DPP is a secreted signal. So secreted signals, they're sent out of the cell and they can diffuse. What prevents it from diffusing down here and then turning on a bunch of, this is going to be your nervous system and your cuticle and so on, your dorsal specific fates. This is mesoderm, it's going to go inside, and this is going to be neurectoderm. So this part, sorry, this is going to be go in and form the nervous system, and this is going to be the ectoderm. It's going to end up around the outside of the embryo as it's developing. So we've got these regions of expression, but some of these proteins, SOG, shortened gastrulation, it can go outside of the cells. Tolloid can go outside. How do these things manage to stay where they're supposed to be? And this is really, really interesting. Okay, so if you go again, the ventralmost part, you've got your dorsal protein in the nucleus, and then in this region here you have SOG. SOG protein is really interesting because it's secreted and it's sitting out here sometimes on, in this region between the cells and the vitelline envelope. And then here's your decapentaplegic protein, and here it comes down the side here. Maybe it's a square. And any time that SOG meets DPP, it binds to it. Okay, so that means it can't signal. It can't go in and into the next cell here and start turning on a whole bunch of genes for the wrong cell type. And as SOG starts to diffuse more towards the dorsal side, wherever it meets tolloid protein, the connection between those two is broken. Okay, and so basically what happened, it's almost like, here's the SOG protein. Whenever decapentaplegic makes it down here, it kind of picks it up, moves it up, and kind of dumps it back off where it's supposed to be. It's quite a brilliant little system. And so the sum total of that, of the three acting together, is that you get lots and lots of DPP activity on the most dorsal region and less on the dorsolateral regions, and virtually none in any of these locations. So this can go ahead and form the neural ectoderm. This part can go ahead and form what's going to be the cuticle on the outside surface of the embryo. And then this is going to be also a source of other signals and things that we aren't really going to talk about, but are really important for making sure the embryo remains intact. Let's see. And yes, as I said, I'm going to suggest, these are the things I suggest, the bonus questions and all that stuff. They're always optional. People will say, do we have to, do them? And the answer is no, you never have to do anything. But I do believe it's an activity that will help you think through and remember how SOG and DPP and tolloid work. And so we'll be talking about that in the class.